Hello everyone. In the ongoing biotechnology series, today I, Dr. Charu Dogra Rawat, Associate Professor at Ramjas College, University of Delhi, will discuss advanced tools and techniques in which I talk about the quantitative PCR and, if the time permits, Sanger sequencing. The learning objectives for today's session would be to understand the principle and procedure of quantitative polymerase chain reaction, to know various applications of quantitative PCR, and to understand the principle and procedure of DNA Sanger sequencing method. Now, when we talk about the quantitative PCR, or as it is called as the real-time PCR, we saw that in the conventional PCR, the amplified DNA product or the amplicon is detected in an endpoint analysis. So, we take a template, we put the primers, we use the Taq DNA polymerase, and it undergoes a number of cycles to make the copies of that template or the gene of interest that we have given, and then we analyze the result on, for example, the agarose gel electrophoresis. So, what we analyze is the end product or the amplified product. But what happens in the real-time PCR? The accumulation of the amplification product is measured as the reaction progresses, that is in real time, with product quantification after each cycle. So, this is real time because as the amplification is happening, as more and more copies of DNA are produced, at each cycle we measure the quantity that is produced. So, it gives us an advantage because then the increase in the copy number can be correlated with the initial template that was present. So, this was a revolutionary technique that came into being. At each cycle during this PCR, the quantity of DNA is measured in real time by using a variety of fluorescent chemistries. Now, how do we detect the increase in the number of copies at each cycle? So, for that we use a variety of fluorescent chemistries. The two methods which we basically use, I will discuss just now. A fluorescent reporter nucleotide in each reaction yields increased fluorescence with an increasing amount of product DNA. So, it basically means that at each cycle, as the copy number increases, the fluorescence, we add a reporter, so the fluorescence will also be increased or enhanced. We detect this fluorescence. So, what we do is we attach these reporter molecules or we add these fluorescent dyes that go and intercalate between the DNA molecules. The specialized thermal cyclers equipped with fluorescence detection modules are used to monitor the fluorescence signal as amplification occurs. So, as apart from the simple PCR machines, the specialized thermal cyclers are used for quantitative PCR, in which there is a fluorescence detection system also, because as the fluorescence signal is produced, as the amplification occurs, this fluorescence detection system detects the fluorescence and gives us the result that how much the fluorescence has increased. Most common approaches to generate a fluorescent signal, they use hydrolysis probes such as the Taqman probes or a double-stranded DNA binding dye such as cyber green dye. So, what are these two methods? The first method we discussed is, for example, the cyber green dye. We talked about while visualization of DNA on the agarose gel electrophoresis that we need to stain the DNA so that we can visualize it under UV light. And if you remember, we used ethidium bromide. So, ethidium bromide used to intercalate between the nitrogenous bases or the stacks between the bases in DNA structure, and when we put it under UV light, it fluoresces and we can detect this fluorescence. So, in a similar way, a more sensitive dye such as the cyber green dye is used, which basically binds to the double-stranded DNA, and it can fluoresce only when it is bound to the DNA. So, on the left hand side, the method is shown here. So, you can see these dye molecules, they can intercalate between the double-stranded DNA molecules. They cannot they do not fluoresce between when they are, because they fluoresce only when they are between the double-stranded DNA molecules. They do not produce any fluorescence when they are in the solution with the single stranded DNA molecules. So at each number of cycle, for example, if we start with one double stranded DNA molecule, after one cycle 2 DNA molecules are produced, and therefore the fluorescence will be doubled. So this fluorescence can be detected. So at each cycle, as the number of double stranded DNA molecules will increase, that means more copies are produced, or our gene of interest or the template amplifies, the fluorescence also increases, and that can then be detected. So this is one of the method that is basically used to assess the PCR product produced in real time at each cycle. There is another method which is called as by using the Tachman probe. So what we use here is what are called as probes. So if you remember, the probes are the ones which are attached to some signaling, signal producing molecules that can produce the signal. So in here you can see they are the locus specific probes. That means they will bind to your gene of interest or the template, and it contains two molecules. One is a molecule, reporter molecule, which is going to produce a fluorescence, and another is a quencher molecule, because it is going to absorb this fluorescence radiation when it is present near to the reporter molecule. So we use these Tachman probes, and these Tachman probes go and bind to the single stranded DNA, just like we use the primers. So when these primers, they synthesize, then due to the 5' 3' exonuclease activity, it is going to remove this nucleotide and continue growing and synthesizing the template DNA. So when it removes the reporter, then it fluoresces, because now it is no more near to the quencher system. So at each cycle, as the primer, which is basically the polymerase which is synthesizing from the primer, it removes the fluorescence is given. So more the number of molecules, accordingly and proportionately the fluorescence will be released and detected. We also talked about certain advantages and disadvantages. The major advantage of this Tachman probe system is that it is very specific. This is there can be non-specific binding, because it is just recognizing a double-stranded molecule and not sure that it is, if there is some kind of a contamination, or even if there is a non-specific template which is there, it can still bind to it. But this is very specific, because the probe is designed to bind to the template DNA only. So there are methods, and we can basically use any of the methods in general, and for common uses the cybergreen method is also used. Now, because we say it as real time, so we understand the meaning of the real time, that while the process is progressing, we can actually quantify the product. So what happens is that we need to quantify basically the product which is produced, and that can correspond to the target that we started with. So an important, and that is why it is called as the quantitative PCR. So an important aspect is the quantification of the product. The qPCR platforms collect fluorescent data from every amplification cycle, and the increase in fluorescence is plotted against the cycle number, resulting in the typical amplification curve. So what these systems do is that they will collect the fluorescent data and then they will plot it against the cycle number, and you will get these kind of plots which are there. So there the PCR is kept with different concentrations of the DNA, the template DNA, as you can see, and then there is an increase in the template product and the detection of the fluorescence signal. So for different products, if the product is more, the curve is different. But the major thing is that the curve will have four phases. The stage phase of the curve would be there. The first is the background one. So there would be a background signal. We will see that it can be produced because of the plastic wear or some kind of a very low amount of fluorescence which will still be there. So that will be the background. As the amplification starts and proceeds, there will be an exponential increase in the template copy number. So still there is an exponential increase, but it will still not be detected because there is a detection limit for the fluorescence. So until and unless it reaches a certain copy number after a number of cycles, this fluorescence will still not be detected. So although it is in the exponential phase, but it will be it will be gradually increasing from the background, but still it cannot be detected apart from the background. So the cycle, the number of cycle at which the fluorescence is detected, that is termed as the CT, which is the threshold cycle, or CQ, which is basically the quantifying cycle or quantitative cycle. So this cycle is the CT value, and then there will be an increase in, on the logarithmic scale, it will be linear amplification, and after a while you will attain there will be a plateau in the fluorescence. That means with the further increase in the copy number, still the fluorescence would not increase, and that will happen because of the exhausting, the polymerase is exhaustive, so there is no more amplification even, and therefore there comes a plateau, no more fluorescence increase after that. Now you can see, when you start with different amount of template DNA, the CT changes. So if you have more template at the starting, then the CT or the threshold will be reached earlier than if you have lesser amount of template at the starting. So this is what we call as quantification. So we can actually quantify that at the start what amount of DNA was present. So if we know that amount of template that was present based upon the CT, so higher the CT, it's basically the larger amount of concentration of DNA or whatever template was present. Lower the CT, then there is a basically the threshold is reached early, so that means there is a higher concentration of template. If the threshold reaches after a while, then there is a lesser concentration. And for example, this is a non-template control. So non-template control, there is also some non-specific fluorescence increase that is occurring, but it is CT is too far off, because it is taking a large amount. The fluorescence will not increase because it does not contain a template only, and this is still increasing because of some kind of a contaminated whatever, or some background that got enhanced, etc., and that is why it is increasing. So this gives a very good method to us for quantifying the initial amount of DNA. So qPCR amplification curve can be subdivided into 4 stages, namely the background noise, where the background fluorescence still exceeds that derived from the initial exponential template accumulation, then the exponential amplification, linear amplification, and a plateau stage. So we actually get this kind of a 4 stages that we had already discussed. Now, what are the reasons for this background fluorescence? The background fluorescence may be caused by a range of factors, which include the choice of plasticware, the remaining probe fluorescence that is not quenched, light leaking into the sample well, and differences in the optical detection for a given microtiter plate well. So these are some of the experimental I would say experimental deviations because of which the background fluorescence would still be there. But that background fluorescence, until and unless the template fluorescence surpasses that background fluorescence, it will not show any increase in the fluorescence. During the exponential phase of the amplification, the amount of target amplified is proportional to the starting template, and it is during these cycles that gave numbers are quantified, the G numbers are quantified using what is called as the CT method. So I just discussed about the CT method. The CT, or commonly called as CQ as well, is reached when the accumulation of the fluorescence is significantly greater than the background level, and we can correlate this CT to the amount of template that was initially present, and therefore we can quantify. During the initial cycles, the fluorescence signal due to the background noise is greater than that derived from the amplification of the target template. Once the CT value is exceeded, the exponential accumulation of the product can be measured. I hope this is clear to you, that how and what is the importance and significance of the CT or CQ value, because on basis of that we can actually quantify the template DNA. When the initial concentration of the target template is higher, the CT will be reached at an earlier amplification cycle. So again, we can do a comparative thing. When we use different concentrations, we observed that if the CT for the sample where the CT was reached earlier, the template concentration was more. So obviously, because the CT will then be reached at a earlier stage. Quantification of the initial target sequences of an unknown concentration is determined from the CT values and can be described either in the relative or in absolute terms. So we can use this unknown concentration of the template. We can derive these concentrations by comparing the CT values. So we can do a relative check. So what happens in the relative check? That apart from the unknown, your template, you also amplify a housekeeping gene, for example. So where the concentration is, you will know that this is the constant, what amount of DNA you have entered, the housekeeping gene. There will be a constant CT values that you will get, and you can compare your concentration with that concentration and you can calculate it. So what happened in relative quantification is that the changes in the unknown target are expressed relative to a co-amplified steady state, what is typically called as a housekeeping gene. For example, in prokaryotes, 16S rRNA gene can be used. However, in prokaryotic system, it is very difficult to get the steady state, but in eukaryotic system, you get a lot of genes which are steady state genes. So you can co-amplify those genes, and because they are steady state, you can correlate the number with the CT values, and by comparing it, because you are co-amplifying it to your unknown concentration of the template can be then calculated. This commonly applied for, as I said, studying eukaryotic gene expression. It is more difficult to apply this method for studying prokaryotic genes where identification of a valid steady state reference gene is problematic. However, I give you an example that probably 16S rRNA gene can be co-amplified for studying the relative concentration of the unknown target gene by quantitative PCR method. What happens in absolute quantification protocols? The numbers of a target gene or transcript are determined from a standard curve that is generated from amplification of the target gene present at a range of initial template concentrations, and then the CT values for each template concentration are determined. So this is a typical method where we use a standard curve, when we extrapolate that on the standard curves of the unknown concentration and get the unknown concentration. So you convert it into a linear regression model, and in the linear regression model you can actually, so this is created for a different concentrations, you will get a straight line, and then you can typically put your unknown concentrations and you can check that CT value, how much concentration it is giving. So it is a typical standard curve and you can calculate the unknown concentration by these curves. Some of the parameters that are also reported here are very important. So a simple linear regression of CT values is plotted against the log of the initial copy number. The CT value of the most, so one point that is to be remembered is basically that the CT value of the most diluted template DNA used to construct the standard curve is at least a log fold lower, that is approximately 3.3 cycles lower than the CT value of the no template control. So when there is a no template control, whatever CT value is coming, then the lowest dilution should be at least 1 log fold lower. So when you are setting up the experiment, you need to consider this point, create a standard curve, convert it into log values and find the unknown concentration of the template DNA that you have. So this is all feeded in the computer, in the software, and it can basically, you can choose which method, the relative method. For relative method, you will have to co-amplify a steady state gene. And for absolute terms, it is going to create this standard curve or use a standard curve that is already created and calculate the concentration of the template DNA at every cycle. So as I said, some of the reported descriptors of the standard curve are such as the amplification efficiency, the linear regression coefficient, and especially the y-intercept value, which uniquely describes the standard curve and indicates the sensitivity of the reaction. The CT value of the NTC and its equivalent value in terms of gene numbers are also given. For example, here it is already shown that this many base pairs genes per liter is basically related associated with the NTC value. So the NTC value is also given, and then the CT cutoff is calculated by subtracting that NTC value, and therefore it is basically this CT cutoff is there. So these descriptors are very important to calculate the unknown quantification of the unknown target template DNA. This is basically how the quantitative PCR or the real-time PCR is utilized, how the quantification happens. The major importance of this or the major application of this is basically to quantify the gene expressions. So when we talk about gene expressions, we are not talking about DNA, but we are talking about RNA. So there the template or the starting material is RNA. So when we are trying to quantify the amount of RNA that is present in a cell, then we can use what is called as the RT-qPCR. Now you can go back to the polymerase chain reaction session and you can see what RT-PCR basically means. So it uses an enzyme which is called as a reverse transcriptase, and this reverse transcriptase convert the RNA into complementary DNA. So the RNA is first transcribed into complementary DNA by reverse transcriptase enzyme from total RNA or the messenger RNA. This cDNA is then used as the template for the quantitative PCR reaction. So directly RNA is not used. So this is a very, very important method. For example, if you want to study two cells under different conditions and you want to see what is the change in the expression of a particular gene, or in a diseased or in a non-diseased state, or probably you have to compare a particular RNA in different cells, for example a liver cell and a muscle cell. So all these kind of gene expression analysis can be done by RT-qPCR, where the total RNA would be prepared from the cells under different conditions, from different locations, or for example, as I said, diseased or undiseased state, the total RNA would be prepared, it will be converted into the complementary DNA, and then from this complementary DNA, whatever gene of interest we are studying, that will be amplified, and at each cycle it can be quantitated. So this quantification at each cycle will correspond to the initial RNA that was present. So if in a diseased state, you can say that whether the RNA is increasing in that DNA state, in the diseased state, or the RNA is decreasing in that state, or under different conditions whether the RNA is increasing or decreasing. So you can use two methods, one is a single step method and another is two-step method. So in a single step or one-step method, you add the RNA polymerase as well as reverse transcriptase, the oligoprimers. You can check the reverse transcriptase PCR mechanism in our previous session, and you can put it only in one tube and do it simultaneously, or you can do it one step by step, where first the RNA is converted into the cDNA and then the primers are added, the quantitative PCR primers are added for quantifying this PCR product. So one step or two step methods can then be used. So we discuss further applications in our next session. That would be all for today. Thank you.